Hello guys, so now we're going to look at the importance of T cells and what happens without T cells and also the different types of T cells. So some questions to keep in mind as we'll answer these throughout the lecture are how are T cells selected and how do the subsets develop, and we'll get to the different types of subsets. So again, what are the roles and actions of these subsets? And we'll also look at a little bit of a case study about degeorge syndrome. So again, more questions to keep in mind. What would happen without T cells? And what about the case of the missing thymus? So some things to keep in mind when we go through the degeorge syndrome is to answer some of these questions and test your understanding. So what happens in the thymus? T cell nurture or T cell death? So the thymus is a good place for a thymocyte to be. Thymic epithelial cells form a network around developing thymocytes, providing signals needed for maturation and survival. The thymus is a bad place for a thymocyte to be. But 98% of thymocytes die in the thymus. There's death through apoptosis, which prevents damage to the thymus. So just keep this in mind. So T cells develop in the thymus, which we know T cell, T for thymus. So this is kind of our overview here. We'll get into more detail, but I'll just go through quickly. So our T cell pre-circuitors commit to T cell lineage following notch signaling and initiate T cell receptor gene rearrangements. So here's our notch signaling. Immature T cells that recognize self-MHC receive signals for survival, and those that interact strongly with self-antigens are removed, similar to B cells. So again, they bind to self-MHC, not good, and they're killed. Mature T cells encounter foreign antigens in the peripheral lymphoid organs and become activated. These activated T cells proliferate and eliminate infection. So here in the thymus, T cell progenitors develop in the bone marrow and migrate to the thymus, where cells complete their development by rearranging their antigen receptor genes and undergoing repertoire selection, which we'll discuss. Mature T cells migrate to the peripheral lymphoid organs. Then these activated T cells migrate to the sites of infection, and are involved in usually kill cells or act as T helper cells. So what do T cells or thymocytes have to accomplish in the thymus? They must productively rearrange their T cell receptor genes so they can express a functional T cell receptor. They need to commit to expressing either CD4 or CD8, which are T helper or cytotoxic T lymphocytes. They must survive positive selection, which we'll discuss later, which selects for useful T cells, and they must survive negative selection, which removes dangerous T cells. So again, how do T cells learn to see this complex peptide self-MHC? This is the role of the thymus, but we'll get to that later. So we'll look at some little bit of a structure of the thymus here. So the thymus is organized to facilitate T cell maturation. So essentially, the anatomy dictates the function and helps with the physiology. So the thymus is organized into two major regions, the cortex and medulla. The cortex is the outer region, and medulla is the central region, similar to many other structures, such as our adrenal glands. So the cortex has immature thymocytes and cortical epithelial cells and macrophages. Got our cortical epithelial cells. You see that there. Again, here's our cortex and our medulla. The outer layer is proliferating immature thymocytes. And we have an inner cortical layer, which is immature thymocytes going through thymic selection. So again, here are thymocytes. Then the medulla, which has thymocytes, medullary epithelial cells, and APCs, which include our macrophages and dendritic cells. Here we can see that here, dendritic cells and macrophages. Again, the cortex is densely packed with rapidly dividing thymocytes, so keep that in mind. Oh, sorry, you can see here, just in case you guys know, thymus is located just above the heart there. So T cells undergo development at different locations in the thymus. So the precursor thymocytes do not express CD3, which we know is expressed in all T cells at some point when they're mature, CD4 or CD8. These are considered double negative. So essentially, they're CD4 negative, CD8 negative. These go through 4 double negative stages where T cell receptor gene rearrangements and commitment to the gamma delta or alpha beta lineage occurs. Next, they go through enter a double positive immature thymocyte stage, where they express both CD4 and CD8. So they can said to be CD4 positive and CD8 positive. These mature to a single positive cell, where they express either CD4 or CD8, and again, sometimes referred to as CD4 positive T cells, indicating it only has this, and then CD8 positive. These single positive cells exit the thymus if they pass both positive and negative selection processes. So early lymphoid precursors enter from the bloodstream via venules near cortical medullary junction that migrate to the cortex. So in the thymic cortex, we have densely packed thymocytes interacting with thymic cortical epithelial cells. This is where positive selection occurs and potentially negative selection. These survivors migrate from the cortex to the medulla, where they interact with dendritic cells and cortical or medullary epithelial cells and are involved in negative selection. So again, here's kind of this process here. We have our double negative cells, double positive, then our mature CD4 or CD8. And here's that venule we discussed. So some key locations. So here, it's kind of a different kind of figure discussing the whole T cell maturation process. And then again, we see here at the end our functional T cells. So again, this is a good figure to follow along. So this occurs in the cortex and medulla. So this is essentially a graphic depicting the thymus. And then again, remember our T cells, our lymphoid precursor cells come from the bone marrow and enter the thymus, and then they're double negative. So the key stages in development of the alpha, beta, and gamma delta T cells in the thymus, showing T cell receptor gene rearrangements, expression of T cell co-receptors, CD4 and CD8, and locations of positive and negative selection. So this essentially sums up the figure. Selection begins after T cell receptor genes are rearranged by screening our double positive thymocytes in the cortex, which again, double positive means expresses both CD4 and CD8, and our single positive thymocytes in the medulla. So again, here's our single positive. So positive selection occurs in the cortex. Again, thymic selection. Here's our positive. Negative selection can occur in the cortex or the medulla. The single line at the cell surface represents the CD3 and zeta chains, which again are always expressed on the T cell. So a kind of a way to sum this up is neglect, select, and eliminate. It's kind of a different way to look at it. But this will make sense as we go through T cell selection. So the key cell types involved in thymic selection. So in the thymic cortex, we have our thymic cortical epithelial cells, which mediate positive selection. So again, our thymic cortical epithelial cells mediate positive selection of developing thymocytes and can mediate negative selection. In the thymic medulla, only negative selection occurs. So again, in the cortex, we can have both. That's where positive selection occurs, but we can also sometimes have negative selection. But in the medulla, only negative selection occurs, which is mediated through APCs, including dendritic cells, macrophages, and thymic medullary epithelial cells, presenting self-peptide complex with MHC. So positive selection is the interaction of alpha beta T cell receptor CD4, CD8 double positive thymocytes with cortical epithelial cells. Negative selection in the medulla is due to interactions of single positive thymocytes with thymic antigen-presenting cells and medullary epithelial cells. Here's our negative selection, and here's our positive selection, but some negative selection. So positive selection. So the first question these thymocytes have is, do you recognize our self-peptide MHC molecule? So these thymocytes, which are double positive, interacts with cortical epithelial cells expressing our class 1 or class 2 MHC and peptides. So if the answer is no, these thymocytes die by neglect. So we need them to be able to recognize our MHC in order to function. So this results in no survival signal, and there's apoptosis, which occurs for 95% of most of thymocytes. But if the answer is yes, for those lucky few, these thymocytes survive and they receive these survival signals. Then they pass positive selection. So again, positive selection occurs in the thymic cortex. Thymocytes with T cell receptors able to bind self-peptide complex to self-MHC, including class 1 and class 2. But note, however, that very high binding affinity can lead to cell death. So we don't want too strong, but we just want just the right binding affinity. So interactions during positive selection. The thymic cortical epithelial cells are critical for mediating positive selection. Class 2 MHC interactions with CD4 are needed for CD4 positive T cell maturation. Class 1 MHC, again, is involved with CD8 for CD8 positive maturation. There's a similar approach for class 1 gene expression, which shows their interaction with CD8 is needed for positive selection for CD8 positive T cells. So here essentially are steps. So both CD4 and CD8 positive T cells mature if the class 1 and class 2 MHC expressions is normal. Disrupted class 2 gene expression inhibits CD4 and T cell development. So class 1 will be unaffected because this is for CD8. This restores class 2 expression on thymic cortex epithelial cells, or TECs, which needs to be sufficient to restore CD4 positive T cell development. If the thymic cortex epithelial cells express mutant class 2 proteins unable to interact with CD4, this inhibits CD4 positive T cell development. So here we have a normal MHC class 2 expression. Both of these mature. MHC class 2 negative mutant, only CD8 T cells mature as they use MHC class 1. So a mutant MHC class 2 transgene expressed in thymic epithelium. Again, both will mature. If we have a mutant MHC class 2 transgene expressed that cannot interact with CD4, again, only CD8 will mature. So now we have thymocytes that can recognize their self-MHC. So this is our passing positive selection. So why is negative selection needed? So negative selection occurs in the thymic medulla and to some extent in the cortex, but mainly in the medulla. So the questions we pose now is, do you react strongly with self-peptide MHC? So in positive selection, we need them to react to self-peptide. But now we're testing how strong. So the answer is yes. If it's too strong, thymocytes will die. So high affinity interaction of T cell receptors with presented self-peptide. If there's a strong signal sent, the cell dies. If the answer is no, the thymocyte survives with its weak/moderate affinity interaction, and this weak signal means the cell survives. So remember, this is just testing the MHC. So in our actual system, we have those other co-stimulatory signals as well. So this kind of makes up for this lack of strong binding that we have now. But this is a preventative measure against autoimmunity. So essentially, negative selection eliminates self-reactive T cells. So negative selection eliminates self-reactive T cells. Self-antigens are presented to dendritic cells or macrophages, or thymic epithelial cells to thymocytes. So we see here, T receptor that can bind to self-peptide MHC complexes, but does so with low affinity. This will proliferate, survives negative selection with its weak moderate signal. T cell receptor with high affinity for self-MHC or self-peptide has a strong signal, dies during negative selection. So this high affinity binding, the thymocyte binds strongly to the self-peptides presented by self-MHC, and we have a strong signal transduction via T cell receptor and CD3. This thymocyte dies. Low affinity binding, i.e. the thymocyte binds weakly to self-peptides presented by self-MHC. There's a weak signal transduction via T cell receptor and CD3, and the thymocyte survive. So, summary of this thymic selection process, and MHC restriction and central tolerance. So why is positive selection important? T cells unable to interact with self-MHC are removed, and this results in MHC restriction. Why is negative selection important? T cells that react too strongly with self-peptides are removed. T cells that react to your proteins are dangerous and can be autoreactive T cells. Removal results in central tolerance. Again, here's a good figure to summarize the whole thing. So here we have a positive selection taking place, no interaction, die. They interact with our cells like they're supposed to. On our thymic dendritic cells, that they survive. Then in negative selection, if there's too high affinity interaction, there's deletion, intermediate to weak interaction, survive, then our single positive cells will proliferate. So affinity model of T cell selection. So again, here's the model right here. So death by neglect is initial interaction of the thymocytes T cell receptor with self-peptide self-MHC complexes on thymic epithelium is needed for survival. So that's our positive selection in a sense. So our positive selection, these low affinity interactions of T cell receptor with self-peptide self-MHC on thymic cortical epithelial gives that survival signal. These positive selected thymocytes are screened for strong reactivity in negative selection, where high affinity interactions of T cell receptor self-peptide in the MHC results in T cell death and clonal deletion and self-tolerance of the mature T cell population. We also can have agonist selection, which you can see here. So agonist selection generates T regulatory cells, which are very important. So T regulatory cells are a subset of positively selected cells receiving signals slightly weaker than those inducing negative selection. So they're not completely overpowering, but they're still stronger than the ones that were negative selected for clonal proliferation. So these proliferate into regulatory T cells or Tregs. So the final stages. So T cell maturation in the medulla leads to changes in responses to T cell receptor signaling. There'll be proliferation instead of apoptosis. Mature T cells leave the thymus to enter circulation. There will be thymic emigration, requires the recognition of S1P, which is sphingosine 1 phosphate, by a receptor of S1PR1. This S1PR1, you can see here, is expressed on T cells during final maturation stage. So SIP, we can see here, S1P is high in the blood and lymph, and it acts as a chemoattractant to mature the lymphocytes. Then we have our mature T cells, which also express CD62L, which is an L-selectin, and this directs homing of lymph nodes, similar to what we've seen in neutrophil rolling. So here we see our CD4, CD8 T cells entering the medulla, which are not fully mature. Here's the blood vessel. After 3 to 4 days in the medulla, the CD4 and CD8 T cells upregulate SIP1 and exit the thymus into the blood. And again, using L-selectin, homing to the lymph nodes. So the goals of the thymus are accomplished. There was productive rearrangement of the T cell receptor genes and expression of the functional T cell receptor, both gamma delta and alpha beta. The alpha beta T cell receptor and T cells commit to expressing either CD4 or CD8. They must survive positive selection, which allows for useful T cells to be selected for and expanded in number, and they must survive negative selection, which allows for dangerous T cells to be removed. Or there's agonist selection, where they become Treg cells. So where do all these self-peptides in the thymus come from? So one important thing we'll look at is AIRE, which is called autoimmune regulator gene. So we kind of call this AIRE, or just AIRE. Either one is fine. But yeah, this is kind of gives it away its function in the name, autoimmune regulator gene. So this is a transcription factor controlling the expression of self molecules, aka PTAs, by thymic medullary epithelial cells. So PTA is peripheral tissue antigen, and there are over 500 peripheral tissue antigens. Example would be insulin, myelin basic protein, C-reactive protein. So essentially, AIRE in the thymus essentially presents a bunch of our self-antigens. So like again, insulin. So again, we don't want self-reactive insulin, which is seen in type 1 diabetes, myelin basic protein, which is seen in other diseases, and CRP. So these are presented to the T cells in the thymus. And essentially, if they're too self-reactive to these things through a negative and positive selection, they're removed. So we want them to have tolerance to these things. So, and again, we can have defects in this, which is not good. So if you have a defect in ARE, or autoimmune regulator, there's increased autoimmune disease incidence. We have a disease called APCED, which is autoimmune polyendocrinopathy candidiasis ectodermal dystrophy syndrome, long name, aka autoimmune polyglandular syndrome, and results in isolation of gene responsible for this defect, led to the understanding of ARE role in the thymic selection. So essentially, unfortunately, for some, I guess, was that the understanding this disease led to the discovery of these functions, which gave us more insights. But yeah, it's really interesting. If you want more information about this, check out this link for a paper. So as you mentioned, there's DeGeorge syndrome. So the case of the missing thymus, so that kind of gives it away its function. It's the pathology. So individuals with DeGeorge syndrome, at least those who survive, experience a greatly increased incidence of autoimmune disease. So, a hint, tolerance. Through tolerance, we can prevent autoreactive T cells from causing harm. So just to clarify, DeGeorge syndrome is when we have missing or very little of a thymus gland. So therefore, we have very little T cell function, if at all. So tolerance is when several different mechanisms occurs through several different mechanisms. There's central tolerance, which is induced in the thymus, where T cells or in the bone marrow for B cells. An example is clonal deletion by negative selection. We also have peripheral tolerance, which we talked about, including clonal energy and our Tregs or regulatory T cells. So here we have an example of a Treg functioning in suppressing an autoreactive T cell. So suppression of autoreactive T cells by regulatory T cells requires them to interact with the same antigen-presenting cell. So, a hint. So T cell subsets. So these are pretty important, I would say, in a lot of pathologies and a lot of diseases. This is a really good figure to get down. So I would say the best way to remember is the subset itself and the signature cytokines and mainly the function. So these you can kind of get away with, but still important. But I would say these two are the most important. So our T cell subsets and signature cytokines. So T helper cell 1, or just Th1, which we'll refer to them as Th from now on, promote cell-mediated immunity. We have interferon gamma, IL-2, macrophages, NK cells, CD8 T cells, and B cells, the IgG3. Again, cell-mediated immunity. Th2, which promote antibody production. We have IL-4, IL-5, IL-13, which stimulate eosinophils involved in parasitic infections like worms, and is also a response to allergens. Releases IgE and IgG4. Th17 are pro-inflammatory responses and have been linked to several conditions, but are also very important. They release IL-17, 22, stimulate neutrophils and epithelial cells, and pro-inflammatory, and respond to fungi and extracellular bacteria. T regulatory cells release TGF-beta, IL-10, and other lymphocytes, and inhibit functions of other T cells, and again, regulatory role. And we'll go over these again in more detail. So, what does DeGeorge syndrome tell us about the thymus? So again, here's that figure. Why do individuals with DeGeorge syndrome have higher rates of autoimmune disease? So, this is most relevant for incomplete DeGeorge syndrome, where you still have some thymus, but not all of it. So again, you have some thymic tissue and some T cells mature. But there's problems because we have lack of negative selection process due to the partial thymus. So not all autoreactive T cells are removed, and there's failure of central tolerance. This lack of regulatory T cell development results in the failure of peripheral tolerance as well. So not only are you immunocompromised, essentially, you're having autoimmune diseases at the same time. So again, central intolerance is induced by the thymus through clonal deletion by negative selection, and a peripheral tolerance is through regulatory T cells and clonal energy. So another view of our T cell subsets. So again, our major subsets are Th1, Th2, Th17. We also have TFH, which are T follicular cells, as we can see here, which are very important and probably the more recently discovered in our T regulatory cells. So this is a good figure for more of the functions, doesn't talk about the cytokines as much. So a lot of times we'll have people will refer to as we have a Th1 response, a Th2 response, a Th17 response. So again, if you go back to this figure here, in a Th1 response, we have these cytokines, these cells, and these effects. But here, we can see the main functions and pathogens targeted. So they kind of work together. So Th1, again, macrophages, infected macrophages provide help for B cells in antibody production, and usually involved in microbes that persist in macrophage vesicles and etc. As we talked about, IgE and the activation of eosinophils for parasites. Th17 are very pro-inflammatory and mainly involved in extracellular bacteria and fungi. And TFH cells are involved in all types of pathogens. And then again, our T regulator cells suppress T cell response when we need it. So again, essentially another figure dictating the same kind of thing. So remember, CD4s work with MHC class 2. So we have our Th1 cell bind with APC, releasing interferon gamma. There's also cells that, specific Th1 cells, basically specific cytokines that they produce, but also cause to stimulate. So again, signature cytokines, those are IL-4, IL-5, IL-13, stimulating our eosinophils, and again, helmless parasites. Th17, IL-17, IL-22, stromal cells, and then we can have producing further pathways where we have GCSF, antimicrobial peptides, mainly stimulating our neutrophils, again, highly pro-inflammatory, with extracellular bacteria and fungi. TFH cells, releasing IL-21, helps with mainly functions in isotype switching. Tregs, lack of T cell activation. So again, Th1, cell-mediated immunity. Th2, antibody production, causing antiparasitic responses. Treg, downregulatory role. So CD4 positive effector T cells specialize in different actions that allow them to control the immune system. The T follicular helper cells help B cells promote antibody production and class switching. Again, Th17 are pro-inflammatory responses. Th1, Th2, and Tregs also produce cytokines that can regulate B cell class switching. So importantly, each of these will stimulate the B cells to certain immunoglobulins. So it's not just random. They kind of follow their own specific pathways and specific, as we saw over here, we can see specific immunoglobulins are produced. But we'll get to that later. So, where do these cells come from? T cell subsets are basically released because of cytokine influences. So the CD4 T cell differentiation into different subsets. So even though these produce certain cytokines themselves, they need to be, so like example, we had this T regulatory cells releasing its certain cytokines, Th1 releasing its certain cytokines, and etc. But these themselves need to be stimulated by certain cytokine subsets. And as we'll talk about, these are mainly stimulated by our innate lymphoid cells. So those cells we talked about a lot earlier can kind of follow the pathway. So we have obviously 1, 2, and 3, and luckily the names kind of follow the pathway. So this Th1, Th2, and then this one unconventionally is Th17. But we'll talk about that later. So transferrin growth factor beta stimulates FOXP3, TGF beta, and IL-10, T regulatory cells, and then the rest is what we're talking about here. So then we get to these stages, T regulatory cells. So essentially you could place this figure on top of this figure up here. Then we have IL-12 interferon gamma, T-bet, IL-2 interferon gamma, Th1 cells. IL-4, GATA3, IL-4, IL-5, Th2 cells. So again, this is one where you just, I would say, come back, look at the figures, even draw out the figures. Just draw out your own pathway, going from each step to the next one. Kind of make like a little pathway chart, best way to remember. So again, remember T cells need C3 signals. So we have our T cell receptor, which recognizes antigenic peptide MHC presented by APCs, and our CD3 and CD4 co-receptor induce intracellular signaling processes. So remember 1, 2, 3. We have our co-stimulatory signal, which is delivered by the same APC through our CD28-B7 interaction. So this third signal is where we get that, what do we do next, essentially. So here we get a bunch of different cytokines. This is not an all-encompassing list, but here's an example of many cytokines. So then we have subsets of effector T cells, which depends on the third signal from the APC. So cytokines control T cell differentiation, and these different T cell subsets interact with other cell types and choreograph the immune response. So these antigen-presenting cells, as we know, can be our DCs, macrophages, and B cells. So how does it know which one to produce? It's essentially based on our ILCs, but even that is based also on our what pathogen we encounter. So essentially, our body's luckily smart enough to know that certain pathogens will induce certain cytokines. So like we talked about parasites. So we can have IL-4, which then will stimulate our Th2, which will go down and stimulate more IL-4, IL-5, and then we can see have our eosinophils and our IgE to fight that parasite. But this is all because we had that parasite in the first place. And either we had first initial activation by ILCs, or we detect these can dictate what the pathogen can dictate what we actually happen. But yeah, again, key to remember is the 3 signals needed. So I recommend going through this again, 3 signals, and then you can go back to our last lecture if you need more detail. So these cytokines control the T cell differentiation, like we mentioned. So different T cell subsets interact with other cell types, and again, choreograph this response. So essentially, this is a similar figure we saw earlier. So these cytokines direct the CD4 positive T cell differentiation into different effector subsets. So again, we have our fate-specifying cytokines. As we said, IL-4, Th2, interferon gamma, IL-12, Th1. So these further release these cells as well. So this is where we now have our Th17, and these are the signature cytokines above here. So IL-6, which you'll see a lot in the literature, stimulates T follicular helper cells. We have our TGF-beta, IL-2, Treg cells. So essentially, this just sums it out. IL-12 stimulates Th1, IL-4 Th2, TGF-beta, IL-2, Treg. TGF-beta plus IL-6 plus IL-23, Th17, as we see here, IL-6, T follicular cells. So again, one of those things where if you just, make a little figure, basically make your own little figures here, it'll be easy to remember. Maybe once or twice, and then you'll have it down. So, as we said, we have these fate-specifying cytokines. Here they are. Then these have specific transcription factors and other factors that end up in these cells. So it's not just an immediate thing. They don't just see it and change by themselves. So these cytokines specify T-cell differentiation patterns by activating certain transcription factors, like we said. There are distinct STAT family transcription factors, which you can see all of them use STAT in this stage. And these are activated by cytokine-induced signaling and act to direct T-cell subset differentiation. So Th1 requires STAT1 and STAT4 activation, which causes T-bet expression. Th2 requires STAT6 activation and GATA3 expression. T regulatory cells require STAT5 activation and FOXP3 expression. And STAT3, we can see here is involved in 2, is also involved in Th17 and T follicular cell differentiation, and this is sustained by RR gamma T expression, which is needed for Th17, and this requires IL-23 and TGF-beta. So again, this RR gamma, we can see here is different from PCL6. So again, we'll start with the activated Th2, Tregs, and our T1s. So our Th1s produce interferon gamma. Interferon gamma downregulates Th2 proliferation. So essentially, when we dictate the pathway, so we're going to Th1. As we can see, there's interferon gamma, and this also blocks the other pathway. So again, when we said we can have Th1 responses, or Th2, which we'll talk about, which is important in allergies and stuff like that, it inhibits the other one from responding. So it kind of self-promotes its own pathway. So we can see this represented in this figure. You can see there's inhibition of the other pathways when one is selected. So, and when Th2 secretes IL-4, this IL-4 inhibits Th1 differentiation. IL-4 acts indirectly by binding IL-12, production by macrophages, and this is important because by blocking IL-12, this is one of the cytokines needed to go down the Th1 response, and it needs IL-12. Th1 and Th2 cytokines can also downregulate Th17 differentiation and actions through interferon gamma and IL-4, which inhibit Th17. Our Tregs can downregulate everything, essentially, because that's their role. And so TGF-beta inhibits Th1, 2, and 17 differentiation and activities. And it also produces IL-10, which downregulates the actions of other subsets, which can be produced by other T cell subsets under certain conditions. Again, take out the figure if you want to see. So again, interferon gamma acts on Th2. Sorry, that's a big line. IL-4 and interferon gamma inhibit Th17. IL-4 acts to inhibit Th1. So now the next topic is going to be Th1 and CDL, but we'll see that later. We'll have a whole lecture dedicated to Th1 since it's quite important. So again, here's a subset reminder of the functions of each of the cells. And remember, we have our T follicular helper cells, which are important for B cell help and antibody reduction and class switching. So we'll take a look at some Th2 stuff right here, or in next class. So that'll be 2 lectures from now. So again, we'll focus on Th1 individually and Th2 by itself. Essentially, this is because, Th1 is involved in so many immune responses, while Th2, again, is important for parasites, but it's also heavily involved in certain allergies. So as we discussed, eosinophils and mast cells. And here we can see we'll have IgE. But again, this can be a real big problem and some hypersensitivity reactions, but we'll get to that later. So remember, Th cell subsets and the roles. So these Th2 cytokines, which we'll focus on later, produce cytokines that support B cells and class switching to IgE. These support anti-parasitic responses, but again, as I mentioned, have a role in allergic reactions, including type 1 hypersensitivity. These are both, Th1 and Th2 are both extremely heavily researched, and we'll take a look at them individually later. And I hope you enjoyed it. Take care.